Myths and Misconceptions about Microscopy, sponsored by the Microscopy Lab.
Oh hi, welcome to the Microscopy Lab. You're just in time. This is the first of a series on light microscopy. I'll be covering only a portion of the field of microscopy because it has expanded so much. We will be covering both the theory and some practical aspects of light microscopy use, with special attention to analytical and forensic microscopy, or particle characterization and identification. In this first webinar, we'll break some of the myths or misconceptions of microscope use. We will name parts of the microscope along with an overview on how to align and properly set up microscope illumination for a biological microscope so you obtain the best images possible for your sample. Unfortunately, the internet has a lot of misinformation about this. Some of the topics to be covered include light and optics, an introduction to crystal morphology, different kind of microscopes with an emphasis on polarized light microscopy. We'll discuss dispersion staining, chemical microscopy, and other topics related to the identification of particles from a wide range of industries as well as the world around us. What kind of industries use the microscope? Let's take a look.
What kind of industries use the microscope? Here's a list of just some of the industries or sciences where the microscope can prove to be useful. Not only geology and forensic science, but in obtaining intelligence against terrorism. In pharmaceutical sciences, where polymorphism of a drug can mean differences of absorption into the human body or even effectiveness of the drug itself. Many sciences and industries use microscopy. If you want to learn more about microscopy, nearly any aspect of microscopy, the McCrone Research Institute is the place to go to learn microscopy. You'll learn in a small classroom setting with friendly, knowledgeable instructors using research-grade microscopes, learning theory and practical applications of microscopical methods. These are hands-on the microscope courses that are up to a week in length. And if enough people from a company or region are enrolled in a course, the instructors can teach at your facility. So to learn more, contact the McCrone Research Institute and be sure to check their website for more information. I would like to acknowledge former Cerritos High School teacher Sid Rogers, who helped create the original presentation. Also, I would like to thank Donald Moorhead, videographer and post-production creative assistant, and Lisa Tunnell, videographer. This is the first presentation in a series on the use of the microscope, particularly analytical or forensic microscopy topics. In this presentation, we discuss the myths or misconceptions many students and scientists have about using the microscope. The purpose of the presentation is to begin to teach and educate on the use of microscope by pointing out some of the myths taught to or misunderstood by students from our educational school system. I want to teach the proper use of the microscope and cover lesser known microscopical methods or techniques to expose the student, as well as the scientist, additional analytical tools, as well as improve analytical approaches to problem solving. Some of the images remain the property of the original copyright owner. These images are being used for teaching, scholarship, research, and educational purposes. All rights are reserved to the appropriate owner. Why, I would submit to you that the microscope is the most recognized as well as the oldest scientific instrument. Various forms of simple microscopes have been around since the 1200s, or shortly before the first wave of Black Death hit Europe. And the compound microscope, or biological microscope, was established around 1600, or shortly before the Mayflower landed at Plymouth Rock. Use of the analytical microscope not only provides a very useful tool, but a whole different approach to analytical problem solving. Publications on general science subjects use microscopical images on their covers. Chromatography science journals use the images of crystals with a polarizing light microscope, also known as the PLM. And specific topic journals such as analytical toxicology, use images of crystals with the PLM. This is an example of a replica of a Van Leeuwenhoek microscope, or it's possible that it's the real deal. That would have been from the mid-1600s. This is an example of a simple microscope. It has only one lens. Brian Ford, the world's leading authority on the Van Leeuwenhoek microscope, has taken photographs of microscopic objects using actual Van Leeuwenhoek microscopes from the collection of England's Royal Microscopical Society. He has shown how the single lens microscope by Van Leeuwenhoek provided very good resolution, even compared with 20th century microscopes. We should cover definition of some terms and nomenclature so we know the names of the parts of two common types of microscopes, the stereo light microscope and the biological light microscope. Both of these are compound microscopes, but often people refer to the biological microscope as a compound microscope. We'll cover nomenclature of other microscopes in future webinars. First, I'd like to point out I'll be referring to the words particle, specimen, or object frequently and interchangeably. A particle is any solid substance made of any material, natural or manufactured, of any morphology or shape that is capable of being observed with a microscope. For instance, a pollen grain, fiber, or a quartz sand grain are all particles and are tiny concentrated bundles of one type of chemical able to be viewed with a microscope. We will cover some nomenclature or the names of some parts of two types of microscopes, the stereo light microscope and the biological light microscope. These are two examples of a zoom magnification stereo light microscope, or SLM. They look a little different, but they have the same parts. Some are just in a different place or might look a little different. We will go into greater detail about naming various parts of the stereo microscope in the webinar on stereo light microscopes. Here is some terminology, though, to get us started. At the bottom of the microscope is the base of the microscope and the stage. The stage and stage plate, the round circle you see in the stage, is where you place the object or specimen to be examined. You should always have your specimen on a piece of weigh paper, butcher paper, filter paper, a petri dish, or some other kind of container, and not rest the specimen directly on the stage plate of the microscope. You could contaminate the specimen or your stage plate, or the stage plate could become scratched or damaged depending upon the specimen being examined. The objective, a fixed magnification lens, magnifies the specimen to obtain the first image into the magnification housing, where the magnification changer increases or decreases the magnification of the first image. Of course, you're the one that's actually doing the increasing or decreasing of the magnification using the magnification changer. When the magnification is continuous, it is called zoom magnification. The focusing knobs allow the lens assembly to move up and down, allowing you to put the image of the subject into good focus. The lenses you look through at the top of the microscope are called eyepieces, or also called oculars. I will be using oculars and eyepieces interchangeably. They fit into a part at the top of the microscope head called ocular tubes. Next, we will name parts of the biological light microscope, starting with the illuminator or light source. In this microscope, the illuminator is at the back of the base of the microscope and it is attached. This is still considered built-in illumination. The basic portions of the microscope are the base, the arm, and the head. You should only transport a microscope holding it with two hands, one by the arm and one underneath the base. On the left and right side of the arm of the microscope are the focusing knobs. On the front of the microscope is the on-off switch, and on the right side is a voltage slider, voltage regulator, or rheostat that controls the intensity of the light that the bulb emits. Starting at the light source or illuminator where the bulb is located, the light passes through the base of the microscope and then through the field diaphragm, the condenser diaphragm, and the condenser, and passes through your sample microscope slide, which is resting on the stage. This has a mechanical stage where a slide can be inserted for movement and viewing. Fortunately, the mechanical stage can be easily removed for more efficient movement of your slide. The light from the specimen is collected, corrected, and magnified by the objective, passing into the head and through the ocular tubes of the microscope and then the oculars, where the image is again magnified. Because light passes through two sets of lenses, it is often referred to as a compound microscope. However, technically, most stereo light microscopes have two sets of lenses also. And now, on to the myths. There are 10 myths or misconceptions about a microscope and microscopy that I believe should be corrected. The first myth or misconception is, don't touch the microscope. We have examples of this everywhere from elementary school through graduate school. In many biology-related courses where use of the microscope is needed, no to little instruction is given, and sometimes that instruction is wrong. Unfortunately, with today's emphasis on molecular biology, many high schools no longer even use microscopes. The reality is that the first thing when approaching a microscope is to reach for and touch, then use the focusing knob to adjust the focus for your eyes, as well as to be able to look through the sample. An important adjustment for binocular microscopes is the interpupillary distance. That is, the distance between the pupils in each eye so they are centered to look into the eyepieces, also called oculars at the same time. This distance must be corrected for each person in order to see the object or specimen with both eyes.
With this stereo light microscope, in order to clearly see a single image, you must adjust the distance of the eyepieces or oculars to match those of your pupils. Do this by adjusting or moving the ocular tubes as you look through the oculars until you see a single image. So it's important to adjust the ocular tubes to match the interpupillary distance of your eyes. Some microscopes, unfortunately, do not have this feature. While viewing the specimen with your right eye, use the focusing knob on the microscope to focus on the specimen or a portion of a specimen. Then, while looking at the same specimen or a portion of that specimen, use your left eye and adjust the ocular so that it is now focused using your left eye on that same specimen or portion of the specimen. Now your microscope is adjusted for your pupillary distance and each eye is focused on the specimen. This should mean that you're going to have less eye strain and hence less fatigue while using the microscope.
You can see already how important touching the microscope is for proper use and to maximize your observations with the microscope. Myth number 2. I've seen and heard this many times, just get light through your oculars and you'll be fine. The reality is, after focusing and adjusting the pupillary distance, a microscopist will adjust various parts of the microscope to obtain the best image possible. Here we see the condenser diaphragm, and the field diaphragm. Each has a different purpose and importance in obtaining the best visible image. The proper setup is called Kohler illumination after the microscopist who invented it. To give an example of a microscope that is not properly aligned, the image on the right is in focus, but the condenser is not correctly placed. The condenser is not focused on the specimen as seen in the diagram on the left. The blue lines in the diagram show the ray tracings of light through the condenser and specimen, showing that not all of the light is entering into the objective. You can see that the focal point of the condenser is below the specimen, thereby not providing proper light to the specimen. The punctae, or tiny dots, in the diatom are not visible, but merely implied in this fuzzy image. In this slide, we see the image on the right. The puncta, or little dots, are clearly visible inside the diatom. The focus of the objective did not change, only the condenser position. The condenser is correctly focused on the specimen, and the condenser diaphragm was not adjusted. Only the position of the condenser relative to the specimen. The blue lines in the diagram on the left show the light focusing on the specimen due to correct placement of the condenser. The light from the specimen then enters into the objective. I'll be showing a quick way to achieve this in just a little bit. In another webinar, we'll cover setting up a microscope in more depth. Myth number 2 corollary. Use the microscope in whatever condition it is in when you receive it. No, the type of microscope must be taken into account and the microscope properly aligned or have proper illumination to obtain the most information from your sample. The parameters of your particular microscope must be maximized to observe the best optical phenomena. This slide shows only a few of the possibilities. But in this one slide, we have dispersion staining, orthoscopic observation with crossed polarizers, phase contrast, conoscopic observation, which allows viewing interference figures, and orthoscopic observation with cross polars and a full wave compensator. You'll learn more about these in future presentations. Each of these microscopical observations require a slightly or very different microscope construct by the microscopist. Myth number 3. Turn the microscope light to the highest setting for the best illumination. Students and associates have complained of headaches and tired eyes after using a microscope for a mere half hour to an hour. And when I checked their instrument, in many cases, they had maximized the intensity or brightness of their light. With the microscope set up properly, you need only a fraction of the maximum light to achieve the correct brightness. Note that in each of these microscopes, very different illuminators, one in the base of the microscope on the left, and the other on the right is exterior to the microscope. The voltage is only a portion of the total available, with the light providing ample intensity. To help preserve bulb life, always start with the bulb setting at its lowest voltage or intensity. Then adjust the brightness upwards to between 1/3 to 1/2 to start when first setting up the scope. As you adjust the microscope for proper illumination, turning down the intensity may be required, or you may need to increase the intensity slightly due to the type of sample or the type of microscope setup that is being used. Myth number 4. Begin with the highest magnification. Beginning with the highest magnification, you have no clear idea what you are observing or the matrix around the particle, which could be significant in the identification of the particle and the interpretation of the results. The reality is the microscopist starts at a low magnification and, if needed, proceeds to higher magnification. Here we see the original particle and other particles around it. This appears to be from a construction site or other location where apparent drywall, wood, hot glue fragment, small rocks and minerals, possible insect parts, fibers, apparent metal fragments, and other materials, as well as hair, would be found. Starting at a high magnification, the sample may be forced into the objective when incorrectly focusing and damage the objective, and more importantly, lose your sample in the slide breakage. It's dangerous and a mess to clean up all that broken glass. Here you see the results of damage to the front lens of a high magnification lens to a microscope.
We'll be more specific in another webinar on the details of setting up Kohler illumination. But now, I'm going to give you some highlights on setting up Kohler illumination. Assume the following. Your microscope light is on. The substage and field diaphragms are each about half open. The 10 times objective is in the optic axis of the microscope. You have a sample on a slide on the stage of the microscope under the objective. And now you begin by focusing on your sample. As you focus slowly onto the specimen, move the slide back and forth slightly to more easily see where your sample is. This sample has moderately heavy contrast and is easy to see, but a less contrasted specimen may be more difficult. Without touching the focus, move the slide to a clear area through the mounting media and under the cover slip. Reduce the field diaphragm to its minimum opening. Using the condenser focusing knob, use the condenser to focus the leaves of the field diaphragm in the field of view. Use the centering knobs on the condenser to move the condenser so the field diaphragm is in the center of the field of view. Once in the center of the field of view, you may need to refocus the condenser on the field diaphragm. This may take several tries to get it perfect focus. Condenser is too high, you'll see orange. If it's too low, you'll see blue. Next, you'll want to open the field diaphragm to just outside the field of view. Bring the specimen back into the center of the field of view and begin to adjust the substage condenser diaphragm to get the best contrast and resolution for your sample.
Myth number 5. The stage clips are your friend. Stage clips restrict movement of the sample or microscope slide. Here we see 2 different types of stage clips. Often these have been inserted into designed holes in the stage of the microscope to hold the slide or specimen in place. To remove them, you usually pull them straight up or lift them up and away from the stage when there's no slide or sample present. Some may require a tool such as a screwdriver, Allen wrench, hex driver, or other type of tool. Myth number 5 corollary is that the mechanical stage is your friend. The mechanical stage is a device that moves the slide on the stage for you. When you learn to use your hand to hold the slide while scanning the slide, you can quickly find your particles or search the slide much faster than you can with a mechanical stage. The advantage to a mechanical stage is that it has rules in the X and Y direction, and these rules can be written down to tell someone else where to locate something on your slide. When using the manual method, you'll learn shortly, it is faster than using a mechanical stage. So the best thing to do is to remove the stage clips and the mechanical stage from your microscope stage and use your hand to move the sample around or the slide. So we're going to have now a session on how to remove the mechanical stage.
Here we have a biological microscope with a mechanical stage on it. First thing after putting your slide into the mechanical stage is now you've got to move your mechanical stage to get the slide to where you want to start viewing things. And you only have an X or Y view here in order to view things. So we're going to remove the slide, set it aside. We're going to remove the 2 screws for the mechanical stage. They just come right off. We'll set the stage off to the side and we'll next pick up a microscope slide with a sample on it. To use the stage and the slide and be able to very quickly maneuver our slide around on the stage. And again, you can move it very small increments or very large ones. You can make it so that it's in various different orientations if you need to, unlike your mechanical stage. Clearly an advantage to having it hand manipulated rather than using a mechanical stage.
Myth number 6. Don't move the specimen or microscope slide. The reality is the specimen is only on one place on the slide. Dust and other artifacts exist in many places. You move the slide to find your specimen. After a little practice, manual movement of the slide is quicker, easier, and nearly as exacting as using a mechanical stage.
So, how do you move the slide if you don't have a mechanical stage once your stage clips are gone? I'm glad you asked. When I'm using a microscope slide, I will generally grab it with my middle finger and my thumb to control it. And you can do it this way, or you can use it this way where you're grabbing the entire length. Sometimes for better control, it's better to have it close to where your sample is. With a little practice, you'll be able to move the slide a few microns at a time. Notice how my little finger and my ring finger and my whole hand sort of cup around the stage, and I'm using the middle finger and thumb in order to move the slide. Not always do we use a 1 by 3 inch slide or a 75 millimeter by 25 millimeter slide. Sometimes we use half slides. And even with the half slide, you still use your thumb and middle finger to control the slide on the stage. The same applies with the hand. It wraps around the stage, and you use your thumb and middle finger to control the slide, as you'll see now. Here you can see with the thumb and the middle finger, you have good control on the slide and can move it very small distances on the stage. And this is going across the 1 by 1 inch. You have good control of the slide. The hand still wraps around the stage and allows you to move the stage and observe your particles, while your other hand is on the focusing knob.
Myth number 7, close one eye tight for the best view. Not every microscope has the ability for binocular viewing. Some allow only one eye to be used. These are called monocular microscopes. If you're using one, learn to keep both eyes open. Your eyes will not tire, and you'll learn to see with a single eye for viewing your particles. Also, when using a monocular microscope, switching between eyes once in a while will also help to reduce fatigue. Some microscope users use only one eye with a binocular or trinocular head. Though it takes a little practice, you will avoid a great deal of eye strain if you train your brain to only see what is coming into one eye while the other is still open. Myth number 8, whatever is brought into focus must be what you're supposed to see. Funny how a piece of lint looks very different under a microscope. Since you are not allowed to touch the microscope, whatever is in focus must be what you're supposed to see. On the left are dust or non-relevant artifacts. On the right are the particles of interest. Myth number 9, the only thing you can do with a microscope is to look at tiny objects. In addition to the morphology or general shape of a particle, there is the color in reflected as well as transmitted light, the texture of the surface, inclusions, or some kind of object or pattern internal to the particle that is being observed, the luster or how shiny or dull the sample appears, whether the sample is made of a single particle called homogeneous or different kinds of particles or heterogeneous, or if the size of the particles are similar or different, called micrometry. For some samples, like natural fibers, diatoms, starch, or pollen, morphology and size may be all you need to identify the particles. For others, you may need additional optical properties like refractive index, birefringence, optic sign, and an interference figure to determine a 2V angle. Or you may need to use additional features, such as melting points, or perform microchemical tests that can be used to help characterize and identify the particles. Many of the features on this and the previous slide will be covered in future webinars. Myth 10, there is no relation between chemical structure and observation. Few question the relationship between structure and observation of cells examined for medical purposes, but may have issues with non-medical particles and cells. Here are two references describing relationships between optical characteristics and the chemical structure, but these aren't the only references or research into this topic. This is one chapter out of the Manual of Mineralogy described in the Crystallography and Mineralogy of James D. Dana and updated by Klein and Hurlbut. What we see here is the result of double refraction, or the breaking up of light into two separate rays vibrating perpendicular to each other and each carrying information. By using a special filter called a polarizer, we can allow only one of the two images of the word calcite to be seen from the calcite crystal. The filter permits only the light vibrating parallel to the filter to pass, while blocking the other ray from passing through the filter. So the observer only sees one of the two images by using the filter. In a later webinar, we'll explore more about polarized light and how to use polarized light in the identification of particles. The vibration of the two rays here and how we use this will be covered. There are theoretical constructs of the atomic arrangement within the crystal, giving rise to the passage of light and hence the optical properties exhibited by that chemical arrangement that allows a particular material to be characterized and identified. The Shannon, Fisher et al. Research group has shown the structure and arrangement of atoms and molecules in crystals significantly affects the light transmission through the crystal, resulting in the observed refractive indices. They have compiled a large database of compounds, calculated the refractive indices from atomic measurements, and compared these same compounds with their measured refractive indices, showing the difference in most instances to be less than 2%. Another research group calculated refractive indices from the X-ray diffraction patterns and compared these values against the measured refractive indices on a microscope. Clearly then, there is a relationship between composition, structure, and refractive index. So in review, for myth number 1, the reality is you have to touch the microscope to at least focus the microscope. For myth number 2, the reality is that the microscope must be properly adjusted to obtain the best image for the type of microscope being used. For myth number 3, the reality is turn the microscope light to no higher than 1/3 to 1/2 max. Adjust for the intensity during or after setup and as the specimen requires. For myth number 4, the reality is start with the lowest magnification and increase as needed. For myth number 5, the reality is you should remove stage clips or mechanical stages to more quickly and easily move the slide around on the microscope stage. For myth number 6, the reality is that you must move the sample container or microscope slide to find your specimen, examining the particle in context. For myth number 7, the reality is keep both eyes open when using a binocular or monocular microscope to reduce eye fatigue. Myth number 8, the reality is, in addition to searching the slide, you have to focus on the specimen plane to find the specimen. For myth number 9, the reality is that there are a variety of physical, optical, and chemical tests and lighting techniques that will help you to characterize and identify the particles. For myth number 10, the reality is the chemical structure within the crystal or particle gives the particle its unique properties for characterization and identification. We hope to see you next time in the microscopy lab.
Thanks for watching. To learn more, click subscribe here. And if you'd like to email me at the address here. Until next time, go out and observe something with a microscope.